Now I'm going to show you how to use the LAS software to acquire images on the computer. So, by the end of the video, you will learn how to acquire single images both in the bright field and fluorescence mode. You will also learn how to acquire time frame images, so multi-time images. And so by the end of the video, you will also learn how to acquire movies of your samples. To use the software, so to acquire images or movies of your sample, so we're going to use the user one. That doesn't require any password, so you just click on that. Then you end up with the desktop, and the software is here, LAS v4.6. So you double click and the software will start. So the first thing you would do is to choose where you want to save your data, because all the users will be using the same session, so they have left all the settings from the previous session, so you have to redo the settings. So you go to browse here, and then there is here the desktop. So either you save all your images on the desktop and then before you leave you put them all into your folder, or you select from the list the folder where you want to save them. So let's say that we're going to my folder here. So I would go to Niklas. Then I would create a new folder for today by using this create a new folder button. So let's just call it test. And then to select that you want to save all your data in this folder, you would select this button, which is the set capture location, and that will become red. So all the pictures today will be saved in this folder. So that then you know. Then you go back to the acquire set, the acquire mode, and here again these settings will control the camera settings, and they will have the settings from the previous user. So make sure that you change them again to whatever settings you want to use. So you can adjust the exposure time. You can adjust the gain. So usually for bright field you can put that to 1. The saturation will change the colors of your images. So if you want to have neutral colors, you go to 100. And the gamma changes the contrast of your pictures. So again, the neutral one is 1. If you go above 1, it will emphasize, it will make the dark region darker. So for fluorescence, it can be useful. And if you go lower than that, it will make the dark regions less dark. Image format. So you can have a different format for the live images or for the capture images. So usually people don't change that so much. But I don't know if you want to have a very fast live image, but then a very high-res capture image, you could, you could change these settings. But the color, sometimes people change that. And because they want, I don't know, their fluorescence to be the right color. So if you want to have like neutral colors, the colors your sample should be, either in bright field or fluorescence, you should be here at the center of this wheel. So you press reset and it will be at the center. And you should be in the saturation at 100. And then you will have the proper colors. Then in the mic one here, you can change what objective you're using to have the right scale bar on the image, knowing that there is not the 63. Personally, what I do is that I just acquire one image with one objective, then I know the scale bar and I change it on image A, knowing which objective I'm using. So you may want to change this to have the right scale bar on the image, but you don't need to change anything else. So once you have your camera settings set and you are at the focus, you want to make the light go not to the eyepiece anymore, but to the camera. And so for that, you need to pull this out. And now the light is going to the camera, and that's why you can see your image on the screen. Now, the image may be not bright enough. So then you have two options to change the brightness of your image. You can change the exposure time here. Increasing the exposure time will increase the brightness of the image. You can change the gain. That will also increase the brightness of the image. Let's leave it to 1 for now. Or you can just change the intensity of the lamp. So this wheel controls the intensity. So if you close it, the intensity decreases. And if you turn it towards you, the intensity increases. So these are the 3 ways to control the intensity: the wheel that we just saw, the exposure time, or the gain. Then you see that the image here looks pink, which is not the right color. It should be white, black and white here, more or less, for the sample we've loaded. So you need to do a white balance. So for that, you can press on this button, which will do the white balance on the whole image. So you just click on that, and then these are the real colors. To improve the contrast, you can decrease this diaphragm. This will basically improve the contrast of your image. If you decrease this, it will improve the contrast of your image, which will become also darker, so you would have to increase at the same time a bit the intensity, but then you have a better contrast. So here we were on the 20 objective, so let me just put 20. So the scale bar changes automatically here. If you want to see the scale bar on the image, you have to go here, and you go in, and you tick show scale bar and annotation. If you untick it, the scale bar is removed. So let's show it for now. And then you can go in scale bar to change the settings of the scale bar. So whether you want it at the top, bottom, what font you want, what thickness, what, yeah, everything, settings, colors and everything, the background color and everything. And you can either tick or untick merge all. If you tick merge all, it means that when you're going to acquire an image, the scale bar will be on the image. But if you want to have all the image without scale bar and add it yourself, like using other programs, you just untick that. The scale bar is still visible on the image, but after acquisition, it will not be on the image. And then you just click here, acquire image, to capture your image. So you click, the image will be captured, and it will be stored in your folder where the location we just set. And then you can keep moving in the sample, acquiring another image. You can rename the image here, changing the name. So if you want to acquire either a movie of a process or an image sequence, you have to change the mode you're on, and that is here. So here we were on no sequence, meaning we're just capturing single images. If you want to have either a movie or a multi-time sequence of images, you have to select the right mode. So movie, you would choose that for very short movies, like below 5 minutes, because it takes a huge amount of data, like gigabytes. But if you're going to acquire a process that takes much longer, like more than 5 minutes, you would rather use the multi-time tool, and this will allow you to set a sequence of captured images every seconds, minutes, or hour you want. So you click on here, and you will have either no sequence, which is the mode with just a single image, multi-time, or movie. So let's click for now on multi-time. So there is a new thing appearing here. So there is not just the mic and the camera. The camera, again, is to set the camera settings, and you can still acquire single images. But then there is this thing that has appeared where you can set the sequence of image you want to save. So you can choose to acquire one image every, let's say, 10 seconds. So you press 10, this is seconds, minutes, hours. Let's say we want 300 images. Then there is the previous sequence here that you need to discard. So by pressing here, the bin, and then you just press plus to add the sequence you just set. So 300 images every 10 seconds will make about 50 minutes. And then you just press acquire time-lapse to start the acquisition. So this will start the acquisition, and you see the images will appear one after the other every 10 seconds. And you can pause it or stop it. And here is always showing the last image. So let's stop it for now. And they will all be saved again in the same location, in a folder, in a separate folder called multi-time. Okay, if you want to acquire a movie, you go in the movie. And here you will have this thing appearing, which is going to be a continuous movie. And then you just press acquire movie, and it will save a continuous movie for how, as long as you don't press stop, it will acquire the movie. So now if you're moving the sample, this will be acquired in the movie. Then you can pause the movie and resume it a bit later if you're changing, for example, the filter for the fluorescence or whatever. And you can start it again, and it will acquire a separate file, and you can stop the movie. So this is how you acquire images, time-lapse, and movies of your sample. Some general remarks to conclude. Remember the first time you open the software to choose the location of the folder you want to save all your images by going to Browse, so all the images will be saved there. Remember also to reset the settings on the color wheel so that your images have the right colors. And last thing, remember to change the objective in the software so that the scale bar on the image is the right scale bar of the sample. So now you know all about acquiring images, time-lapse, and movies using the LAS software. And thanks for watching and work safely.